Hello everyone and welcome to this video. In this particular video, we are going to talk about the amino acid properties. So there are general properties of all amino acids. So first they are amphoteric in nature and have both acidic and the basic group in them. So the acidic group is the carboxyl group and the basic group is the NH3 group. So they are joined to the peptide bond and they release a water molecule. So also they have a different properties due to the different R group. So these different R group determine the properties of the proteins that contain those amino acid and dictate what a protein can and cannot do and how it folds. So the different property of every amino acid it's due to this different R group. And all these 20 amino acid have both 3-letter and the 1-letter abbreviations. So this is the amino acid chart which we are going to refer. So first we consider the aliphatic amino acid, which is the this one, the glycine, alanine, valine, leucine, methionine, and the isoleucine. Also proline is included into it. So coming to the glycine, so the 1-letter abbreviation is the G and 3-letter abbreviation is the GLY. So in general, the first 3 letter of any amino acid is considered as the 3-letter abbreviation of that amino acid, except for the isoleucine, where I is considered from the iso and the Le considered from the leucine, and the 3-letter abbreviation is the ILE. And for rest of the other protein, it's a first 3 letter. So coming to the glycine, so the R group here is the hydrogen. So due to which it is the achiral molecule and it is found in a flexible part of proteins. So coming to the valine, leucine and the isoleucine, so they are very much similar to each of it. So they are considered in the one group. So they are extended aliphatic chains and they can be branched. And coming to the methionine, so methionine single letter abbreviation is the M and the 3 letter is MET. So they contain sulfur and can interact and bind with the metal ion and often found in the metalloproteins. And now coming to the proline, it is the amino acid rather than the amino acid. So it affects protein folding and they are often found at a bend in a protein 3D structure. So proline is also known as the helix breaker. So the glycine and proline are also known as the helix breaker, and the hydroxylation of proline is important for the structure of the collagen. So the proline hydroxylation is very much important for us. So proline is therefore essential for us. So coming to the aromatic amino acid, so these are the phenylalanine, tyrosine, and the tryptophan. So this group is the aromatic amino acid. So these are the bulkier group. So coming to the phenylalanine and tryptophan, so there are fluorescent molecule. They have a UV absorbing capability at 250 to 300 nanometer, and they can be very useful to identify protein in a mixture. So the tryptophan is converted to serotonin. So serotonin is a neurotransmitter. So serotonin has a sedative effect. So it gives us a pleasant feeling. So very low level of serotonin is are associated with the depression, and the extreme high level produce a manic state. And also tyrosine, it is also a fluorescent molecule and it absorbs UV light at the 280 nanometer, and they can be phosphorylated due to the OH group present into it. So if we see a tyrosine molecule here, the R group contain a OH and this OH can be phosphorylated, and it is derived from a phenylalanine. So phenylalanine hydroxylase convert phenylalanine to the tyrosine, and it is converted to ketocolamines, which include the epinephrine. Epinephrine, also known as adrenal hormone, which is also known as fight or flight hormone. It causes a release the glucose and other nutrients in the blood and stimulate brain function. So the tyrosine, serine, and threonine, which contain the hydroxyl group, so they can be phosphorylated. So the tyrosine contains OH group, the threonine contain OH group, and the serine also contain the OH group. So these three amino acids can be phosphorylated, and they are involved in the signal transduction pathway. Now coming to the neutral polar amino acids, so these are those amino acids which are having a polar group but doesn't have any charge on them. So these are the serine and threonine, asparagine, glutamine, and the cysteine. So the polarity is for the serine and threonine, the polarity is contributed by the hydroxyl group. So the sugar attached to the serine and threonine to form a glycoproteins. So it form a O glycosylation. So the serine and threonine can have a phosphate attached, and they regulate the some of the activity of some proteins. Now coming to the asparagine and glutamine, so they are classified as amides So they are neither acidic nor basic. So they forms hydrogen bond due to the polar groups, and the asparagine can be modified with sugar to form a glycoprotein. So the asparagine linked sugar are the N glycoproteins. So coming to the cysteine, it is a sulfur containing amino acid. So methionine and the cysteine are the sulfur containing amino acid, but only cysteine can form the disulfide bond. So the sulfhydryl chain gives the polarity, and this cysteine can be modified by the addition of the farnesyl or the geranyl groups. So example are these are requirement for the membrane association of the RAS protein. So they can be oxidized to form disulfide bond, they strengthen protein structure, and the disulfide bond are covalent, but they are reversible upon reduction. So the methionine here, it is having a sulfur group, and the cysteine also contain the sulfur group. So this sulfur cannot take part in the reaction, but this cysteine can take part in a reaction. So when the two cysteine molecule form a disulfide bond, that is called the cysteine molecules. Now coming to the charged amino acids, so first we'll consider the acidic amino acids. So glutamate or the glutamic acid and the aspartate or aspartic acid are the acidic amino acid. So they are negatively charged at physiological pH. And coming to the basic side chain, so the basic side chain are positively charged at physiological pH. And the lysine, arginine and histidine are also positively charged at the physiological pH, but only histidine is ionizable near physiological pH because it can act as both proton donor or acceptor depending on the pH in the local environment. So 50% is protonated under physiological conditions. So there are some general rules which we all need to remember. So charged hydrophilic residues are ever buried tend to cluster on the outside of the protein in water. So the charged molecule or the hydrophilic or the polar amino acid are basically found on the surface of the protein, and the polar residue are usually found on the surface, but they can also be buried. And the inside of the core of protein mainly contain the non-polar or the hydrophobic amino acid. So the non-polar residue are also found on the outside of some of the protein. It's not that the hydrophobic amino acid will always be in the core, but they can also be found in the outside of the core or the at the surface of the protein. And the recognition of one biological molecule by another is done by the charge, polar and the non-polar interactions. So I hope you get the idea of the amino acid properties, and I thank you for your patient listening, and like, share and subscribe. Thank you.